Assalamu alaikum. This is our last lecture in cell biology, in the cell biology part of this course. We'll be talking about the biology of cancer cells. So let's move on. So what is cancer exactly? Basically, it starts with the formation of a tumor. And tumor is any abnormal proliferation of cells. So it's basically a mass of cells, كتلة of cells. And these cells have a potential to become cancer. So a tumor is like a benign collection of cell cluster of cells in a certain place. Now they can become malignant, that is cancer, when they invade surrounding tissue and when it spreads throughout the body, and it can use the circulatory or lymphatic systems to do so. Now, the way cancer develops is basically complex, and it depends on the cancer type, and even within the same cancer, like breast cancer, colorectal cancer, and so on, there are different mutations. And we're talking about the key word, the key term, is that cancer develops as a result of accumulation of mutations. This is sort of like simplification of the process. So it's really a multi-step process. And each stage requires certain types of mutations. And these mutations provide the cancer cells with the ability to proliferate uncontrollably, to invade, and to metastasize. So, for example, this is an old idea from the 1990, before you were born, and it was developed by these two people, Fearon and Vogelstein, from Johns Hopkins University. And basically, they said at that time that in order for colorectal cancer to develop, you have it goes through several stages, biological stages, and each stage requires a certain mutation. So, for example, you have mutation in the beta, in the E-cadherin, beta-catenin pathway, um, resulting in the development of adenoma, benign tumor. Then you have mutation in the RAS oncogene, resulting in uncontrollable proliferation of cells. And then you have several types of mutations as well, including P53, and that results in the development of carcinoma and then invasive cells, invasive cancer cells, and metastasis. So that's the idea. You have accumulation of mutations. تراكم طفرات جينية. Now, cancer is really old. Okay, it's not really something new. And cancer was discovered in the bones of dinosaurs, actually. And this is an example, so you can actually go back and read this in the Science magazine, the Science journal. The basic idea behind how cells become cancers is that they are exposed to carcinogens. And there are two types of carcinogens: initiators and promoters. The initiators are factors that cause genetic mutations, making cells susceptible عندهم القابلية to become tumor cells. So, but being exposed to an initiator is not enough. And the initiator can be radiation, pathogen, some pathogens, viruses, chemical carcinogens, and so on. Now, in order for them to become aggressive, cancers, that is, malignant, they have to be exposed to promoters, which are factors that stimulate cells to grow and proliferate. Now, as they do, they accumulate mutation. Now, this is the key term. accumulation of mutations. Now, some of these mutations can be random, and some of these mutations can give an advantage to cells to become cancerous. For example, they become resistant to therapy, they can become mesenchymal-like, they can become invasive, and so on. So you need an initiator and then followed by a promoter. Now, some of these promoters can be certain chemicals, hormones like estrogens. For example, estrogens can promote mammary cells, uh, breast cells in women to become cancer. Certain pathogens, viruses, smoking, by the way. Smoking, the chemicals in smoking, by the way, contain both initiators and promoters. Okay, and that's why those who smoke have a propensity, they have a higher probability, a higher chance to develop cancer. So please stop smoking if you do. So let's emphasize this point a bit further. So we have normal cells right here. Okay, let's say we have a mammary gland, normal breast cells. Mutations can take place in different types of cells. Again, uh, by being exposed to an initiator or by chance. Now, some of these cells can grow, and as they grow uncontrollably, they accumulate mutations, and these mutations create a heterogeneous mass of cells. Some of these cells, they differ. Okay, some of these mutations can give advantage to certain cells. Eventually, what happens is that they can metastasize. Some of these cells can metastasize. They can go somewhere else, and they can develop a tumor mass somewhere else. And that is the main cause of death from cancer. It's not really the formation of tumors, rather it is the spread of tumor cells or cancer cells to different sites. So what are the biological features of cancer cells? One of them is clonality, meaning that they develop from certain cells, single cells, and then these cells can form a tumor. So they are clonal. That is, they all come from certain cells, single cells, and these cells just develop into a tumor and eventually cancer. They have uncontrolled proliferation. They keep on growing. Now, sometimes, by the way, it takes years for cancer to develop. You have accumulation of genetic mutations. Again, this is a feature of cancer cells, and they have this autocrine growth stimulation, meaning that eventually what happens is that some of the cancer cells can secrete growth factors that act on the cells themselves. That's autocrine signaling, uh, in stimulating cells to proliferate. Something else as well is that they have loss of density dependent inhibition, meaning that they keep on growing. Okay, so normal cells grow, and once they reach a certain density, they stop growing. But cancer cells do not stop. Same thing with contact inhibition. That is, once they can contact each other, once they form a contact with each other, they stop growing. So, for example, if you have an injury in your skin and cells proliferate to close the injury, uh, normal cells, once they touch each other, they stop growing, but cancer cells cannot. They keep on growing and growing and growing and piling up. Now, they have reduced cell-cell contact. Eventually, what happens is that you have this cell-cell contact, tight junctions, adherence junctions, and so on. So you have different cell-cell contact points and contact types, and they lose this cell-cell contact. Eventually, they become mesenchymal, they can invade, they lose E-cadherin, they develop or express different type of integrins that allow these cells to adhere to different matrix proteins. Instead of laminin, for example, they would adhere to collagen. Now, what happens then is that cells would invade, they would migrate and invade. They move from their original site, and they move through the tissue, reaching blood vessels, and then they can spread. Now, as they move, of course, they adhere to different types of proteins. They can form focal adhesions so that they can, crawl. They can, and they have to degrade the matrix proteins to enable them to migrate. Now, other features include their ability to induce angiogenesis. Genesis is creation. Angio is blood vessels, is basically the formation of new blood vessels so that they can feed themselves and they can reach the blood easily so that they can spread. They lose differentiation. So for example, here you have a normal prostate cells right here. Okay, so they are well differentiated, and they are functional. Now, once in one of it's progressive, right? So what happens is that these normal cells lose their differentiation. You have what is known as well differentiated adenocarcinoma. Carcinoma is cancer. Um, so you don't have a normal structure of the tissue of the prostate tissue. Then they lose differentiation further, forming moderately differentiated carcinoma, adenocarcinoma, and and they lose their differentiation altogether, as you can see in here. They lose the ability to undergo apoptosis, cell death, so they keep on growing again. They do not die. And the expression of the enzyme telomerase also allows them to stay alive. We will talk about telomerase in the, again, molecular part of this course. So, what are the different inducers of cancer? Well, genetics, of course, mutations in DNA. Certain viruses can cause cancer. We'll talk about some of these. Bacteria, like Helicobacter pylori, the bacteria that causes gastric cancer, cancer in the stomach that causes ulcers, that causes hyperacidity in the stomach. It can cause ulcers. Eventually, they can also this bacteria can also cause cancer, gastric cancer. Radiation, of course, because it induces mutations. Chemicals, certain chemicals, again, chemicals in cigarettes can cause cancer. Environmental factors like sunlight. Okay, sunlight can cause mutations in the DNA. Certain epigenetic factors, we'll talk about epigenetics, so keep that in mind, epigenetic factors. Now, can stress cause cancer? Well, some say that it can by manipulating, by changing the immune system, the ability of the immune system to eradicate, to eliminate, to remove abnormal cells, including cancer cells. How about magic and sorcery? How about envy and the evil eye? Can they cause cancer? Well, if you believe so, you know, you better leave the School of Medicine. So, we'll talk about oncogenes and tumor suppressor genes. We talked about them before. So, an oncogene is a gene that is capable of inducing one or more characteristics of cancer cells when activated. Now, usually these oncogenes they normally exist in our cells. They normally exist in our DNA. And they are called, when they are normal, they are called proto-oncogenes. So a proto-oncogene is a normal gene that can be converted to an oncogene. It can cause cancer if it gets overactivated. How about tumor suppressor genes? Well, these genes are they are needed to control cells from overgrowing, over proliferating. Okay, so they keep the number of cells limited. Okay, so but once they're mutated, once they lose their function, you have cells getting out of control, growing, dividing out of control. Okay, so for oncogenes, we have what we call gain of function. For tumor suppressor genes, we have loss of function. Interestingly, some of the oncogenes originated from viruses. And when these viruses infect our normal cells, these oncogenes can cause cancer. So for example, we have the RAF, the RAS, the SARC, and the AKT genes originally came from viruses. Okay, and these genes exist in our system as well, in our DNA. When they are mutated in our DNA, they can cause cancer. If these viruses that contain these genes infect our normal cells, these cells can become cancerous as well. So now oncogenic proteins can include any of the ones in a signaling pathway that stimulates cell growth. So for example, we talked before about growth factors uh, binding to their receptors, inducing signal transduction, like the RAS MAP kinase pathway. RAS would activate RAF, RAF would activate MEK, then ERK goes into the nucleus and it induces certain genes that would stimulate cell growth. Now, mutations in any one of these factors, mutations that cause overactivation of these proteins, can result in cancer. So over production of growth factors can overstimulate the receptor. Mutations in the receptors themselves to keep them active, mutations that keep these receptors active all the time, can stimulate the signal transduction pathway. Mutations in the RAS protein, mutations in the effectors can also cause cancer, overstimulation of cell growth. Okay, so any one of these intermediates, any one of these molecules can be mutated and can cause cancer. So let's talk about a few examples. So receptors. We have a receptor known as the epidermal growth factor receptor. Now, there is a certain number, low number of these receptors on the cell surface, on the surface of normal mammary cells, breast cells. What happens is that you can have something known as gene amplification. So instead of having a one copy of a gene that would produce the HER2 receptor, the human epidermal growth factor receptor, you have multiple copies of this DNA by an unknown mechanism. We still don't know what the mechanism is that would cause uh, this part of the chromosome, this part of the DNA, to have multiple copies of the DNA itself, multiple copies of the gene. That would stimulate, that would actually cause the overexpression of the receptor on the cell surface. Okay, and that would make cells even more sensitive to the growth factor. And that would cause, again, cell growth, out of control and accumulation of mutations and eventually cancer. So some of these mutations can, in addition to gene amplification, you can have mutations in the kinase domain. You can have mutations in the ligand binding domain so that the receptor becomes independent of the growth factor. Or you can have a deletion mutation so that the ligand binding domain does not exist anymore, but you have the cytosolic part that becomes active even in the absence of growth factors. This is true for breast cancer, and this is why we have this drug known as Herceptin or trastuzumab. It's a monoclonal antibody that would bind to the receptor, inhibiting the receptor, so that cells become, so that this signaling pathway becomes inactive. This is a good treatment, effective treatment of breast cancer. So, like I said before, mutations in transducers and effectors can also cause cancer. So, for example, a certain mutation in the RAS oncogene would result in the production of an overactive RAS, even in the absence of a ligand and an active receptor. So mutation in amino acid number 12 that changes glycine to valine, you don't have to memorize this, but this mutation keeps RAS GTP bound, active all the time, and GTP does not get hydrolyzed. Now, RAF, the effector, the kinase, can also be mutated. So, for example, you can have a mutation in valine number 600. It can be converted to glutamate. Again, you don't have to memorize this mutation, but this mutation can cause an overactive RAF. Similarly, RAF contains a kinase domain and a regulatory domain. I hope you remember what a domain is. A three-dimensional structure that is part of a protein. It can fold independently of the rest of the protein, and it has a functional feature. It's functional. So mutation in the regulatory domain would make the kinase domain independent of the regulatory domain, and it makes the kinase domain active all the time. So deletion of the regulatory domain can make this kinase domain active again all the time. Mutations can also take place in the transcription factors. So for example, we have these two transcription factors that induce the expression of cyclin D1. Mutations in these transcription factors can result in overexpression of cyclin D1, and cyclin D1 would activate CDK4 and CDK6, producing more cyclin E, like I said before, and progression of cells through the cell cycle. Now, in addition to proliferation, so what we talked about before is induction of cell proliferation. Differentiation can also be affected. Now, when we talked about differentiation, we said that when cells differentiate, they lose the ability to proliferate. But what happens, so for example, hematopoietic stem cells can differentiate into myeloblasts, and these myeloblasts eventually can differentiate into granulocytes. Now, what happens is that you can have a mutation diverting differentiation into uncontrolled proliferation. Okay, so cells do not differentiate anymore, rather they keep on proliferating, thinking that that's what they should do. And this is why some researchers think that maybe we can stimulate cells to differentiate instead of proliferating. Okay, so and that would be a good idea of treating certain types of cancer. Survival as well. Apoptosis can be blocked. um, or cells can be prevented from dying. So, for example, activation of the AKT pathway, the PI3 kinase AKT pathway, can keep cells alive all the time. They do not die anymore. So you have inhibition of apoptosis and activation of cell survival. And in fact, mutations in the PI3 kinase pathway have been found to be common in breast cancer and other types of cancers as well. Okay, so cells keep on living, they do not die anymore. Well, that's true for oncogenes. How about tumor suppressor genes? Well, same thing. Mutations in tumor suppressor genes can cause cancer as well. An example, we talked about the PI3 kinase, and we said that PI3 kinase, mutations in PI3 kinase pathway, mutations in AKT can result in overactive signal transduction pathway, leading to increased cell survival, cells do not die, and increased cell proliferation as well. Well, mutations in the tumor suppressor gene called PTEN. Now, PTEN is important because what it does is that it counteracts PI3 kinase. PI3 kinase causes phosphorylation of PIP2 to PIP3, and that results in the activation of AKT. PTEN, on the other hand, is a tumor suppressor gene. What it does is that it dephosphorylates PIP3, converting into PIP2, so that AKT does not get active anymore. Well, mutations in PTEN can cause an overactive AKT, resulting in cell survival and increased proliferation as well, but mainly cell survival. Well, how about mutations in the proteins, the genes that regulate the cell cycle? We previously talked about mutations in cyclin D. We talked about mutations in the retinoblastoma gene that inhibits the progression of the cell cycle. Well, mutations in the inhibitors of the cell cycle as well, like P16, P21, P15, P27. We talked about some of these proteins and what they do exactly. So inactivating mutations can cause, in these tumor suppressor genes, retinoblastoma, the inhibitors can cause cancer as well. How about P53? We talked a lot about P53. And P53, we said that if there are no mutations, P53 is degraded because there's no need for it. But if there is a mutation, or if there are mutations in the DNA, that would cause activation of ATM-ATR, and that would cause phosphorylation of P53. And when phosphorylated P53 can cause either block in the cell cycle progression, or it can induce apoptosis. Okay, by inducing the pro-apoptotic BCL2 family members. Okay, so mutations in P53 are really important in causing cancer. And like I said before, 50% of cancers have mutations in the P53 gene. Now, there are other genes as well, like ATM. We talked about ATM before, but there are two important genes, BRCA1 and BRCA2. Then the abbreviation is given because they cause breast cancer. And they are responsible for a large portion of hereditary breast and ovarian cancers. Okay, so they can be familial. Okay, and these are called stability genes because what they do is that they maintain the integrity of the genome. That is, if there are DNA breaks, these BRCA1 and BRCA2 are activated by the ATM-ATR genes or proteins. For example, ATM-ATR can activate BRCA1, BRCA1 can activate BRCA2, and both of them can be involved in DNA repair. Now, BRCA1, when activated, it can also activate P53. And P53, like I said before, well, it can be involved in cell cycle arrest, um, as well as apoptosis if DNA cannot be repaired. So here we have a connection between the different signal transduction pathways, different molecular mechanisms in the cell. So I said before that some viruses can cause cancer, and they can cause cancer by introducing oncogenes or by inhibiting the function of tumor suppressor genes. And here's an example. We have two proteins that can be produced from the genome of the human papilloma virus. These two proteins are known as E6 and E7. Now, E6 binds to P53 and it causes its degradation. So cells, even if there are DNA breaks, DNA mutations, P53 cannot function. It cannot stop cells from growing. And DNA cannot be repaired. So again, you have accumulation of mutations. E7, on the other hand, would bind to retinoblastoma, blocking it from stopping the cells from going through the cell cycle. Okay, so this is an example. And by the way, the human papillomavirus can cause cervical cancer in women, and it has been sort of treated cervical cancer in the West um, by introducing a vaccine against the human papilloma virus. So the cases of cervical cancer have been reduced dramatically over the past 10, 15 years. And there is a direction to vaccinate females in our own countries like Jordan, Saudi Arabia, um, to prevent human papilloma virus. Alhamdulillah, cases of cervical cancer are not really common in our societies because we are good people, clean. But things are changing. Things are changing in our societies, and there is an increase in cases of cervical cancer, and governments are thinking seriously of vaccinating females. So keep that in mind. The last thing that I want to say is that cancer can be caused by mutations in different genes, and it can affect, and these mutations can affect different signaling pathways. But it seems that some of these pathways are cancer specific, cancer type specific, and even cancer subtype specific. So, like I said, breast cancer, for example, is not really a single type. There are different subtypes of breast cancer. Same thing with colorectal cancer. And it seems that each subtype can be carry certain types of mutations and can have overactive or downregulated signal transduction pathways, certain specific signal transduction pathways and genes. So, for example, mutations in the receptor tyrosine kinases, RAS, for example, are common in certain tumors and even subtypes like colorectal cancer, for example. Okay, mutations in P53 can be found to be common in certain types of cancers as well. Same thing with the cell cycle proteins and the PI3 kinase signal transduction pathways. So mutations are not really mutations that can cause cancer, or certain cancers are not really random like we think. And this is a nice paper. If you wanna just take a look at it, look at the figures, it can give you an idea about what I'm talking about. Allah معكم.